All right, the male reproductive system. So when we're talking about the male reproductive system, we're going to start with the external genitalia of the man. And basically this is split into two categories, the penis and the scrotum. So we'll start with the penis. It is the support structure, so we're going to find our urethra. And it's three main parts. We got the root, which is the attachment point, the body, or the shaft of the penis, and then the glans penis, or the head of the penis. So we look at the body of the penis. What we have inside of it is three masses of erectile tissue. And these masses of tissue fill with blood, and that's what leads to an erection. Now, two of these masses, two of the three are the corpora cavernosa, and these are the dorsolateral masses, so they're along the top and the sides. And we can see in our picture here the corpora cavernosa and how there's two of them side by side. Then the third one is called corpus spongiosum, and that is the erectile mass that actually surrounds the urethra. All right, so upon stimulation, blood is going to fill the spongiosum and the cavernosum cavities, and causing the penis to widen and lengthen. As the tissue widens, the vein that drains the blood out of the penis gets compressed, so blood cannot leave, and an erection is then maintained. All right, so with our penis, we got a couple of ligaments that we need to talk about. We have two that support the weight of the penis and attach it to the front of the pubic symphysis. We have the fundiform ligament, which arises from the linea alba, and then the suspensory ligament that arises from the pubic symphysis itself. And so we can see both of those ligaments in our pictures here, and their attachment points and how they actually both come up and attach to the front of the pubic bone there, and how they are going to support the weight of the penis. The root of the penis, we said, was the attachment point, and with that we're talking about the bulb and the crura of the penis, or the crus of the penis. So the bulb is attached to the deep muscles of the perineum to help anchor the penis in place. Just so we're clear, the perineum is also known as the taint. It's the area in between the anus and the reproductive organs in both men and women, the perineum. So the bulb of the penis is going to pass through the deep muscles in this perineum. The crura extend, these are just extensions of the corpora cavernosum, and each of these attaches to the ileum for further support. So we can see in our picture here the bulb and the crus. All right, the glans penis, the head of the penis. So we call the raised edge the corona. The glans penis itself is the head of the penis. That's where we're going to find all of our sensory receptors. The foreskin, if it's there, is called the prepuce. And then the opening is the external urethral orifice. This little external slit-like opening for both semen and urine. Another picture, this time with the prepuce pulled back so we can see exactly what the corona is. Yeah, let's keep going. All right, the average penis size in men for an erect penis is 5.16 inches with a girth of 4.5 inches. So a little over 5 inches long, 4 inches around is the average size, just worldwide. A lot of men rate penis size to their confidence. It's interesting that 85% of women are satisfied with their partner's penis size. Yeah, almost half of the men feel like their penis is too small. Furthering on that topic, research done showed that only about 20% of women said penis size was important. Only 1% said it was very important, versus 22% that said it was very unimportant. So, fellas, ladies, don't think about it as much as you think they do. All right, the scrotum. This is the support structure for the testes. We have an external median ridge called a raphe. Internally, our scrotum is going to divide into two separate sacs, each containing a single testicle. One testicle will hang slightly lower than the other one does, and that just is to avoid compression, because it's not pleasant to compress a testicle. Alright, so another shot of some testicles there, paired oval glands in the scrotum. This is the site of sperm production. Our tunics of the testes. So we've got two tunics that we want to be familiar with. The first is the tunica vaginalis. This is just a membrane that partially covers the testes and allow the testes to move independently inside of the scrotum. We can see that in the green outline here. That is my tunica vaginalis. The tunica albuginae extends inward and forms these compartments. We call each of these compartments lobules or lobes. So we're using our tunica albuginae to kind of subdivide each testicle into these individual little regions. Alright, so inside each of these lobules, we find a convoluted or tightly coiled tube we refer to as the seminiferous tubule. If we look inside the seminiferous tubule, we got three different major cell types to be familiar with. So we're going inside of the seminiferous tubule to talk about these three types of cells. The first of which is called a spermatogenic cell, or spermatogonia, you may hear it as. These are sperm stem cells, and these are what start producing sperm once they get stimulated. Spermatogenic cells, or spermatogonia, sperm stem cells that start making sperm. Our second type are Leydig cells, also known as interstitial cells, and their job is to produce testosterone. Testosterone is an androgen. Androgen is a hormone that promotes masculine characteristics. So Leydig cells are producing testosterone. Sertoli cells, also known as sustentacular cells, they produce androgen binding hormone, or ABH. Androgen binding hormone, ABH, and our Sertoli cells also form a blood-testes barrier to prevent an immune response. So good little picture showing a breakdown of kind of where we're at so far. We've got the testes, and we can see how the seminiferous tubules are arranged inside of the testes. All right, so with our Leydig cells, they are producing testosterone. Our Sertoli cells are producing androgen-binding hormone. When testosterone and androgen-binding hormone combine, they are going to start promoting our spermatogenic cells, or spermatogonia, or sperm stem cells, to actually start producing sperm. Now, during puberty, sperm production begins in males via signals from the anterior pituitary gland, which is releasing gonadotropins. Specifically, if we think back to the endocrine chapter, we know our gonadotropins are luteinizing hormone and follicle stimulating hormone, or LH and FSH. So our LH is going to stimulate our Leydig cells to start producing testosterone. And remember, this is all happening at puberty. Testosterone then is the androgen, and so that's when young men start showing masculine characteristics. They start to get the facial hair and the body hair. The voice changes all during puberty because of LH stimulating the Leydig cells to actually start producing testosterone. At the same time, FSH, or Sertoli cells, FSH stimulates the Sertoli cells to start producing the ABH, or the androgen binding hormone. And again, when the androgen binding hormone actually binds to the androgen testosterone, that's going to stimulate sperm production in the seminiferous tubules of the lobules in each of the testes. So they're kind of a little summary of what we've covered so far here with our hormones, all on this one slide here. This is a good slide to pay attention to because it kind of breaks it down. We want to associate the L for LH and the L for Leydig cells, and that leaves FSH for the Sertoli cells. Any sperm that are produced now move into the seminiferous tubules, or move from the seminiferous tubules into the epididymis. Epididymis. Little picture there showing kind of where we're at. Let's keep going. So the epididymis, a comma-shaped organ along the upper border of each testes. There's a head, there's a body, there's a tail. Its job to store sperm. Is also the site of sperm maturation. So the sperm, we're storing the sperm and they are maturing while we are storing them. It takes about 14 days for sperm to mature. And it's also going to function to transport the sperm into the ductus deferens or the vas deferens. Now the ductus deferens extends from the epididymis to the ejaculatory duct. It also stores sperm and conveys them to the urethra during sexual arousal. Now, this is also the structure that they cut or tie off during a vasectomy. And this is one of the things that pass through the spermatic cord. You see our nice long ductus deferens there, moving from the epididymis all the way up. So the spermatic cord. Spermatic cord is just a little cord-like structure that's essentially made of fascia, and it just separates the testes and the epididymis in the scrotum from the other stuff in the abdominal cavity. So in our spermatic cord, we are going to find the ductus deferens, which we've already said, but we're also going to find the testicular artery, the pampiniform plexus, which are the testicular veins. We've got the testicular nerve, and then the cremaster muscle are all in the spermatic cord. With our testicular nerve, we want to know that it arises from the T10 spinal segment. With our testicular artery, this is also known as the gonadal artery. And with our pampiniform plexus, it's really interesting. It's a network of veins that are going to drain the testes, of course. But these veins branch and wrap around the testicular artery. And what this does is it cools down the blood before it gets to the testes. And so we can see in our picture there just a little piece of the red testicular artery popping out. But most of it is this blue, that pampiniform plexus. And we see how it branches and completely wraps around and envelops the testicular artery, cooling the blood down before it gets there. And we need to cool it down. Our normal human body temperature is 37 degrees Celsius, but normal sperm production occurs actually at 34 to 35 degrees Celsius. So we need the testes to be 2 to 3 degrees cooler than what our body temperature is. And so another way that we can help control this is through the cremaster and the dartos muscles, and how they can help further regulate the temperature of the testes. So we've got the pampiniform plexus helping, and now we've also got the cremaster and the dartos muscles helping to regulate temperature. Now the cremaster muscle does it by raising or lowering the testes to help regulate temperature. If it's cold, they're going to pull the testes up closer to the body cavity so we can get that temperature where we want it to be. Or if it's hot, they're going to move the testes further away from the body cavity to get to that optimal temperature that we want. Now with the dartos muscle, so the cremaster muscle goes up and down. The dartos muscle, when it's cold, this muscle contracts and it causes a wrinkling of the scrotum to occur, which we can see in the picture to the right. We can also see that the cremaster muscle has pulled those testes up closer to the body cavity. But when the wrinkling occurs, that traps heat in the testes to keep them at that temperature for normal sperm production. When it's hot, this muscle relaxes and it allows the skin to kind of smooth out, and that just makes it easier for heat to escape. So a great little picture here showing everything from the spermatic cord to the pampiniform plexus. We got the testicular artery back there, the epididymis, the testes itself, the prepuce, the glans, penis, the cremaster muscle versus the dartos muscle we can see, and the scrotum. So a great little picture here. So when proper temperature is reached and sperm are being produced, they move from the seminiferous tubules of the testes into the epididymis and into the ductus deferens. So a little review there of where we're at. So we've produced the sperm in the seminiferous tubules, then we've moved them into the epididymis where they have matured, and now we've put them into the ductus deferens for storage. Our ductus deferens extends up and loops over the bladder before joining up with the seminal vesicle. Now the seminal vesicle, it produces fluids that are going to make up about 2/3 of the semen volume. Fructose is the most important component of this fluid that we're getting from the seminal vesicle, because sperm use fructose as an energy source. It's very similar to glucose. So it's a carb, so it's easier to break down for energy production. And the fluid containing fructose that gets produced by these seminal vesicles mixes with the sperm moving through the ductus deferens before moving everything into the ejaculatory duct. The ejaculatory ducts are just formed by the union of the ductus deferens and the duct of the seminal vesicle. So those come together to make the ejaculatory duct. It's going to pass through the prostate, and it empties sperm and seminal fluid into the urethra. The prostate gland is about the size of a golf ball. It surrounds part of the urethra. We get some prostatic secretions that are going to get added into our semen. So we get citric acid, which we could use for ATP production, proteolytic enzymes, specifically PSA, or the prostate-specific antigen, and that's one thing that they measure for prostate cancer. They check the PSA levels. And then we have seminal plasmin, which is just an antibiotic to help decrease bacteria in the semen. And then our prostate secretions are going to contribute to sperm motility and viability, so it's going to help them get there faster and live longer. And it's going to make up about 25% of the semen volume. The bulbourethral gland is also known as Cowper's gland. These are just a pair of little pea-sized glands, little glands. We can see in the picture on the left, there's a little arrow pointing to it and how it empties into the ejaculatory duct. So it opens up into the spongy urethra. It secretes an alkaline fluid to neutralize acids that are from the urine in the urethra. We also use it for lubrication, so it decreases the number of sperm damaged during ejaculation. In layman's terms, this is called pre-cum, but it does have a function. It's there to neutralize any urine in the penis itself and for lubrication purposes. The urethra we talked about in the urinary chapter, so a quick little review here. 8 inches long in men, terminates at the external urethral orifice. We divide it into 3 sections depending on what it is passing through. Prostatic urethra passes through the prostate, membranous urethra passes through the deep muscles of the perineum, and the spongy urethra passes through the corpus spongiosum. Semen, composed of sperm and secretions from the seminiferous tubules, from the seminal vesicles, from the prostate gland, and the bulbourethral gland. All of those components and sperm come together to create semen, or to make semen. Its function, this is the fluid in which sperm are transported. In it, we've got some nutrients for the sperm, and we're going to use it to neutralize the acidity of the male urethra as well as the acidity of the female vagina. We're going to make about 300 million sperm per day. Once ejaculated, they're going to survive for about 48 hours in the female reproductive tract. There are various components. It has the head, which is going to contain the nucleus and the DNA. It's got the neck, which is going to have our centrioles in it. The acrosome is really interesting. It's this little pouch of enzymes that we find on the actual head of the sperm that kind of explodes when it touches the egg, and these enzymes help eat a hole through the egg so the sperm can transmit its DNA mother load. We have a middle piece to our sperm. That's where we're going to get our mitochondria producing ATP so sperm can move. And then the flagellum is actually what's powering the movement in the sperm. So a little picture there breaking down the various components of the sperm. We can see on the head where the acrosome is, so that little pack of enzymes right on the head, ready to touch the egg. So ejaculation, we get expulsion of semen from the urethra to the exterior of the body. Typical ejaculation is 2.5 to 5 milliliters of semen. So we get some parasympathetic and sympathetic nervous system involved in the process. So with erection, we think point. This is parasympathetic, P for point, P for parasympathetic. And then ejaculation, shoot, is our sympathetic nervous system. So S for shoot, S for sympathetic. So point and shoot, parasympathetic, sympathetic, so we can make that determination of what is in charge. And that is the male reproductive system.